All right, so our Bacta incinerator is heated up. You can tell because it's glowing orange on the inside. And then I gathered the materials we need to do this part. So I grabbed our E. coli and our Staph epi. And then these are the TSV tubes. They're pretty much the same color as TSA, and I believe they have the same nutrients as TSA for the bacteria. It's just that it's in liquid form. And so before we begin, I'm going to label my tubes. We're not going to incubate these, but since I'm doing two different cultures, I want to be sure that I can tell them apart. Because once they're in these tubes, I won't. They'll look exactly the same. So this one's going to be E. coli, and then this one is Staph epi. And it's actually Staph epidermidis. So we are at step 5. Use aseptic technique in your loop to transfer a sample of your culture from your plate to the tube of TSB. So we'll re-sterilize our loop for 5 seconds. So 1, 2, 3, 4, 5. And I'll cool it on a blank part of the auger, and then pick up some bacteria culture. Oh. And then with your pinky, you can open the lid and then just transfer it into the liquid. And then recap it and put it back in your rack, and then re-sterilize your loop. So 1, 2, 3, 4, 5. And then I'll cool it on a blank piece of the auger, and then pick up some bacteria culture. I don't know why Staph epi is really hard to pick up for some reason. It like doesn't want to stick to the loop. So use your pinky to open it, and then just put the culture into the broth. Close your culture and then put it back in the rack, and then we'll re-sterilize our loop. So 1, 2, 3, 4, 5. Then we'll let this cool. And then, so now we're at step 6. It says, screw the cap of the TSB down tightly and shake the tube to evenly distribute the bacteria in the tube. So I usually like to do it this way, but yeah, you can just shake it as long as you verify that the caps are closed. All right, so I just quickly wanted to show you this. This shows you how you are supposed to do a lawn. So first you draw the bacteria through the center of the plate, and then you spread it across the plate, and then you spread it across the plate slightly at a diagonal, and then you spread it across the plate at a slight diagonal but the other direction, and then you will go over the sides. Alright, so I just briefly want to go over how you would go about doing a surface swab, which is also called a bacterial lawn. And we'll be using this technique for both the antibiotic disc exercise and for the UV light exercise. So you'll grab a clean, well, whatever media you're using. This is TSA, but for the antibiotic discs, we would be using Mueller-Hinton plates. So first you would go about labeling your plate. So let's just do E. coli, so the dates, and then your initials. I'm gonna grab a piece of paper so that I can better explain what I'm about to do, because when I tried to explain it as I was doing it, it was kind of difficult. Alright, so before I begin to actually do the bacterial lawn surface swab onto this plate, I'll draw it out of what I'm going to do. So say this is your plate. So first, some sterile swabs, and then you open it on this side, so there's like a little crease to open it. But the purpose of opening it on this side is that this is the handle, and it makes it so that the tips that are sterile remain sterile until you pull it out. So you'll saturate your swab with the culture that's in the TSV, and then draw it down through the middle of the plate, and then you will use the swab to disperse it across the entire plate. And then what I do is from the perpendicular angle, you swab it this way, so to move it across the plate, and then I do it from a diagonal across the plate, and then from the opposite corner I do another diagonal across the plate. So when you're done, the entire plate is covered, and so now I will do that onto our plate. So I'll get my E. coli ready and grab the tube. So you want the auger face down so that when you just lift this, you can work underneath it. So we'll saturate our swab. And just place a line down the middle, and then swab it across. Then turn it perpendicular and swab it across. And then from an angle or a diagonal, swab it across the plate. And then from the other diagonal, swab it across the plate. Oh, and then I missed one part. Then afterwards you go around the edge, and then you're done. So you can see the swab pattern that hopefully, that it got the entire surface. And then an example of a mistake that people make is that they either don't do it enough times, or they do the zigzag pattern too openly, so it leaves exposed spots that don't have bacteria on it. So once you're done, this used swab would go into the autoclave bucket, and then you're ready to move on to the experiments. All right, so now we're at step 8. It says sanitize your forceps by dipping them in alcohol Hold the forceps near the opening of the incinerator to speed the evaporation process. Do not insert them into the incinerator. Do not overheat the forceps. You only need to evaporate the alcohol. I'll grab my forceps, which you might know them as tweezers, and then submerge them into the alcohol. And then we just put it at the opening to dry them. And they're almost dry, not quite yet. Almost. Okay, they're dry now. So now we're at step 9. Using your disinfected forceps, retrieve and place a Kirby Bauer disc onto your plate. Into the center of each section marked previously. Use your forceps to ensure the microbial discs lie perfectly flat onto the surface of your plate. Do not push the discs into the auger.